So we were talking about vision, and you learned all the parts of the eye, all the different important things. I'm just going to remind you that the photoreceptors, the actual receptors that pick up on light rays in your eye, are found in the retina in the form of rods and cones. And rods were the ones that served a purpose of giving us vision in dim lighting situations, and also our peripheral vision. The only photo pigment in rods is rhodopsin, just one, okay? The cones were the ones that had the ability to give us fine detail and color vision. That's in our cones. We have 3 different types of cones: red, green, and blue, and their photopigments correspond to the type of cone. So if it's a red cone, it has a red photopigment. A green cone has a green photopigment, and a blue cone has a blue photopigment. Super cool. Did we talk about the cyclic changes and how these photopigments will bleach out and then return again? And that's why we do those like tests, you know, like on Instagram or Facebook, where they tell you to stare at a picture for a long time. And then if you keep staring at it, you'll eventually see something different because you bleached the photopigment in those rods and cones, and then it comes back with a different image. Why are you not with me? What's going on? You're with me. Erica, what are you doing? You're so distracting right now. Okay. Okay. But we did talk about all this, right? Okay. So when it comes to color blindness, because I think somebody had asked a question maybe about color blindness. Oh, no, I don't really want to explain it. Not today. Okay. Oh, the idea of color blindness is not that you're not seeing colors at all, because remember, you have red, green, and blue cones. And so every color that we see is actually a mix of stimulation between red, green, and blue. Those are your main ones. That's it. Yeah, did anybody bring the glasses? Did you bring them? Oh. Okay, then, so you don't deserve to hear about color blinding. You can't even follow instructions, because I had 3 people in this class that are colorblind. Oh, that's so sad. So when you are colorblind, it means that you are missing one of these photopigments. Do you not see color at all? No, you do see color. You just don't see them the same way everybody else sees them. Okay, so like say, you know, orange is like a mix of red and blue and a little bit of green. So if you are missing your red photopigment, you won't see it as orange. You may see it more as a blue or a green. Make sense? Okay, well, that's it. I'm not doing any more with that. Okay, moving on. Fields of vision. So we obviously have 2 eyes, and each eye is going to bring its field of vision into the brain. But how do they do it? Let's talk about this just for a minute. The way the eyes are positioned in our head, notice that they kind of go this way and that way. Each one has its own field of vision. So that would be the field of vision for the left eye, and this is the field of vision for the right eye. Notice that they overlap in the center. We call that binocular vision. It's using 2 different visual fields that overlap to give you one complete picture. That is binocular vision, just like the binoculars that you use or used to use in the 70s or whenever. I don't even know when people stopped using them. So notice these come in on either half. I can only use one finger at a time. Come in through the optic nerve. There are fields that will cross, and then it all goes to your primary visual area in the cerebral cortex. Bottom line, the visual information from the right side of your visual field will go to the left side of the brain. The visual information from the left side of your visual field will go to the right side of the brain. So it's the brain's job to take in those fields, merge them into each other, turn everything right side up and inside out, because remember the images are all reversed and inverted, right? And then process all of that information. And that all happens in the visual cortex. Primary visual area of the cortex. Cool? Right fast. I'm ready to move on. Yes, ma'am.
That'd be so confusing. If you still like saw the world as like upside down or whatever.
It wouldn't be confusing because that would be the only thing you know.
I know, but
right? No, I mean, think about it. That's the world you know from the moment you are born. It wouldn't be unusual.
Yeah, it's like when babies get hearing aids and they cry and then I'm just going.
Yeah. Just like if, you know, at some point in time in the future, if aliens decide to land here, we could be totally bizarre. Our whole world could be upside down to them because their world could be something else. And they're like, this is not normal, right? Your normal is what not even you create, what your environment creates. Just saying. I know there's a little bit of philosophy in there. You're welcome. All right, moving on. Hearing in equilibrium. What? It's Thursday. Ain't nobody getting me down today. Okay, so hearing in equilibrium. Where does that take place? Hearing in equilibrium. In your ear, right, in your ear. Absolutely. And it all comes through either sound waves or movement of fluid. Your hearing is sound waves. Sound waves. Woo. So when you hear something, it goes through your ear, it vibrates that eardrum, that tympanic membrane, and then those vibrations carry on through the middle ear, through all those little three bones, and then it goes on to the cochlea, and the cochlea is the part of your inner ear that has the receptors for sound in the form of vibration. What's really cool is, look at this right here. The ability for our ears to pick up on, to transmit electrical signals from vibration is 1,000 times faster than the response of our photoreceptors to light. So our hearing receptors are 1,000 times quicker at sending impulses to the brain than our photoreceptors are of sending light impulses to the brain. So let's think about that for a minute, okay? What travels faster, light or sound? Light. Light travels faster, right? If you forget, remember, you always see lightning before you can hear thunder. Or maybe you didn't know that. I'm one of those people that will stand outside in a storm and be like, now what is it first? Lightning or thunder first? So yeah. Yeah, that would be me too. So light does travel faster than sound, but our receptors for sound are 1,000 times faster than our receptors for light. So at the end, light's going quick, but the information's traveling slow. Sound is going slower, but the information's traveling quick. They sync up and you get a picture and sound that match. Super cool. That was just a little tidbit of information for you. Don't you love how I give you, this is for trivia night. All right, y'all are welcome. Okay, so the ear has also has our ear receptors for equilibrium. We're gonna see that in the semicircular canals. There are three, I don't even need to go there anymore. There are three parts to our ear. Let's talk about them right here. We have an external ear. What makes up the external ear? Your earlobe is the auricle. That's that part that is cartilage that you put piercings in. The external auditory canal is also part of your outer ear. And then at the end of that canal is a tympanic membrane that you know as the eardrum. So all three of those make up your external ear. What about your middle ear? What's going on in the middle ear? Not a lot. We have three little tiny bones, the malleus, the incus, and the stapes. And then we have this canal right here called the eustachian tube. You may see it referred to as the auditory tube or the nasopharyngeal tube. That is the tube that connects your middle ear to the inside of your mouth, and it's super important because you need that to equalize pressure, right? And if you don't believe me, get a cold or get an allergy and go up in a plane or go down, dive down into the sea. Anytime the atmospheric pressure changes, if you can't equalize it through this tube by having air pass in and out, you end up with a chamber here that's locked under negative pressure. Negative pressure is like a vacuum. It sucks things in. If you suck in that eardrum, number one, you're not going to be able to hear. Number two, it's going to be super painful. Why can't you hear? Because if this membrane is pulled so tight against that negative pressure or towards that negative pressure, it can't vibrate. And the eardrum's just like a drum. If you can't cause a vibration, there will be nothing happening, right? So it's painful. You can't hear. Sorry, and you're liable to have it rupture because it is being pulled in a direction that it doesn't need to go. So that's your middle ear, the three bones, malleus, incus, stapes, and the eustachian tube. Then on the inner ear is right there. Super cool. You got your semicircular canals and your cochlea. Any questions? We're going to talk about details now. Am I talking too fast? What? Yeah, and so that little trick of opening up that eustachian tube is so important. And a lot of people will like travel with like babies and not realize what's happening. And the child is screaming its head off. I'm like, would you please just give it something to drink? It just needs to equalize pressure. I refer to a child as it. Because it is really painful. And us as adults, we know, chew some gum as you're going up or down, you know, or just do that the Salva maneuver where you plug your nose and you blow just to open those tubes up and get that equalization. Yeah. It's also why when you get like a cold or whatever and it closes up, it gets very painful. Your ear can hurt. And then this tube in children is very different. That's why kids always get ear infections, because in adults, it's elongated and it angles down like that. In children, it's shorter and it kind of goes out like that. So it being so level with the nasopharynx, it's really easy to get bacteria in there and have things go up in there and cause an ear infection. So if you have a young child that constantly gets ear infections, don't worry. As they grow and their heads grow a little bit, it will angle down and be a lot less frequent. It just takes time for them to grow into their own bodies. Okay. Are you sure I'm not going too fast? Okay, good. All right, the external ear, we've got the auricle, the external auditory canal, and then the tympanic membrane, which is your eardrum. The purpose of this cartilage, that auricle, is to focus all of those sound waves into that canal, sort of like a funnel. The external auditory canal will transmit those sound waves over to the eardrum at the end of it, and then the eardrum's going to vibrate with those sound waves, and then it goes on to the bones behind it. There are glands in the ear canal called ceruminous glands. We've talked about those because they produce earwax, and I know I've given you my Q-tip lecture. I feel it's an important PSA, and I give it out as much as I can. But those are the ceruminous glands that are producing that earwax. I don't have to do that anymore. Whew. Okay, the middle ear, 3 tiny bones in the middle ear. The malleus, incus, and stapes. So once that sound wave goes through the external auditory canal and it hits the eardrum, it's going to make it vibrate. That vibration goes right on to the first bone, the malleus, which is actually attached to the eardrum. If you've ever had a chance to look inside someone's ear, has anyone ever done that? It's super cool. Ooh, I wonder if I have the camera. I think I have my camera. I think it's in my car somewhere. I don't know. I had brought the camera to show you guys, and actually I brought it because I like to check on my earrings. But anyways, I might have the camera with me. But if you ever get a chance to look at an eardrum, you'll notice it looks like a, at the end of the canal, you'll see the circle, which is the eardrum. And then you may see a tiny little white dot. And then it's so shiny, it should be shiny. It's kind of reflecting light off of that. That little white dot is actually the malleus where it's attached to the eardrum. But it's super cool because you can tell a lot just by looking at that eardrum. So sound vibrations go off of the eardrum onto the malleus, which onto the incus, onto the stapes. The stapes is going to be connected to the oval window, which will transmit those vibrations over to the semicircular canals and the cochlea. All right, we've already talked about the eustachian tube. It's also called the auditory tube or the pharyngeotympanic tube. I prefer eustachian just because that's a universal name for it. I think auditory tube is something new they came up with. Whatever. And it is important to regulate pressure in the middle ear. And here's a nice look. Yeah, see where it pulls it back, it connects it right there, sort of like stabilizes that tympanic membrane. That's the part that you can see when you look in there. Super cool. You can also see if there's like fluid or pus back in there. Anyway, so yeah, that's your malleus, your incus, and your stapes. And then this is your auditory tube. Remember, we want to keep that patent, make sure there's no negative pressure going on. Decongestants are awesome. All right, on to the middle ear. In the middle ear, we have the cochlea. And the cochlea right here, this big snail shell, is the part that had the organ of Corti. Remember that from lab? Don't remember any of this from lab because you wouldn't be the first ones if you don't. Just saying. Every single lecture class, y'all are my last lecture, every single lecture class has not remembered the cochlea or scala vestibuli or scala tympani or the cochlear duct or any of this stuff. I don't know why you guys think you can block it out, and I'm not going to have a heart-to-heart because I've already done that 5 times this week. But y'all, I promise you, AMP1 and 2 are related. Not only that, but what you took in lab is very closely related to what you're taking in lecture. Even if you haven't done it yet, you will get a chance to do it at some point. So please make sure you're not trying to forget lab and understand that they're both tied to each other, even if they're not synchronized. So the cochlea was the part that had the organ of Corti. It had a scala vestibuli, scala tympani, and that spiral organ or organ of Corti that had the hair cells in it was inside the cochlear duct. Remember that? Yeah? Okay. Good deal. And then the semicircular canals are the ones over here that look like a roller coaster, and that's pretty much what they do. They give you those sensations that you have when you're on a roller coaster. Are you spinning? Rotational force. Are you accelerating, decelerating? Like, I feel so bad because I feel like I pour my heart out sometimes, and y'all just don't listen. Like, for real. We talked about this in lab for so long. All right, so this is a slide right here, number 32. If you had this printed out before this week, you might want to check on slide 32, make sure you have the correct one because I did have to go back and edit it. If yours starts off with pressure waves up here at the top, then you have the wrong one. Okay, then just scratch out that part and add this part right here about the vibrations. So sorry, right there. That's the part that's missing. Somehow this got copied twice and I didn't have the top part. Yours is old too? Yeah. Well, the new one's on Canvas. You can just, yeah, just make a note so you don't forget. And it's only, it's like 2 sentences that you got to take. Okay, so vibrations are coming from that last bone, the stapes, onto the oval window, and that's where they're going to go to the cochlea. The cochlea is filled with fluid. Okay, remember, scala vestibuli, scala tympani, and the cochlear duct are all filled with fluid. It's mainly perilymph in the scala vestibuli and tympani, which is very similar to lymph or plasma. So it goes to the scala vestibuli. From there, it's going to go also to the scala tympani. And while it's in the scala vestibuli, it is going to cause those vibrations or pressure waves to pass over to the cochlear duct through that vestibular membrane, right? And it will also cause the basilar membrane, the one that the organ of Corti was sitting on, to vibrate. So basically, I am at this oval window giving vibrations through here that are going to travel throughout scala tympani, vestibuli, and the cochlea. Everything in this little snail is now vibrating. Remember, it's all filled with fluid. When that fluid vibrates, do you remember this slide right here where you had the cochlear duct like that, and then you had an organ of Corti that looked like a pinwheel kind of thing there, right? And then the tectorial membrane just above it. And inside of here you had hair cells. So when those hair cells, when this vibrates and this vibrates, that gets pushed up, makes contact with that tectorial membrane, and that's what initiates that action potential of a sound wave or vibration going through, right? So everything vibrates, the hair cells will go up against the tectorial membrane, and that's what gives you that impulse of sound. And here's that same picture just blown up a little bit more to show you. All right, let's talk about those semicircular canals. If you do remember when we were doing this here in lab, the semicircular canals had these areas, these ampullas, where they had a crista, the mound with the hair cells on it, and all of the hair bundles. Remember that? And it was all filled with fluid. Every time your body moved or your head moved, if that fluid would move, that would initiate impulses from those hair cells. So it's those semicircular canals that help our cerebellum with equilibrium. In particular, it is rotational and acceleration or deceleration is what it is going to pick up on. So, and this is where we in lab had talked about vertigo, you know, where you feel like the room is spinning, right? And we talked about what could cause it and how it's treated. So each ampulla, those little bubbles that you saw at the base of those roller coasters, will have a crista. The crista will have hair cells. When that fluid moves, those hair cells are going to also move in that endolymph, and that's going to give you your action potential. Now, this from the semicircular canals is going to give us that vestibular branch. From the cochlea, we'll have the cochlear branch. The two together will give us a vestibulo-cochlear nerve. This is not new material, I promise. Okay. All righty. And there's that picture that you guys all recognize and remember, right? With the fluid in here. I can't even draw waves anymore, but y'all get it, right? Okay, good deal. All right, so here is a summary of all of that we just did. I will let you guys read over that on your own. Let's talk about aging. Yay. I think I'm going to start omitting this part of all the slides because you don't even have to read it. Okay, let's talk about aging. What happens when we age? Y'all know, right? Everything goes down. Everything goes down. It all goes down. Yeah, pretty much. Yes. All right. So I don't want you to focus too much about the ages here, but it says around the age of 50, your smell and taste are affected. And it's because those receptors are gradually dying off. Your basal stem cells are also dying off in numbers. There's only so many that you can replenish. And so as we age, we will be able to smell and taste less and less. Okay. It says around the age of 40, but I promise you it's not around the age of 40. It is the day you turn 40. For real, it's the day you turn 40. You wake up and you're a different person. I'm telling you right now, okay? What? It's true. It's true. And then it just downhill from there. Every day is worse. So the day you turn 40, you'll wake up and find that your lens has suddenly lost elasticity. Yes. When the lens loses elasticity, it can no longer change its shape, right? Because we saw earlier that the lens can become thicker when you're looking at something close up or thinner when you're looking far away, right? You lose that ability, and that ability to change thickness is called accommodation, right? So if the lens can no longer accommodate, you're going to wake up on your 40th birthday and go out to CVS and buy a pair of these babies. And you will go from the coolest person on earth to the biggest dork on earth because now you got to use glasses to read everything, including a menu at the bar. It's really sad. Some of us like to have salty snacks with their drink. Just saying. Like who just drinks and doesn't have a snack? And now they don't do peanuts and stuff anymore. You have to have like real things. So, and I'm a sucker for beer, cheese, and pretzels. I'm telling you right now. And House of Henry has some amazing ones, but if you go late at night, yeah. So anyways, yes, your lens does lose elasticity. You do lose that ability to accommodate. You will need reading glasses, and I'm sorry, it's not fun. In addition to that, the muscles of the iris, like any other muscle in your body, will also begin to weaken. And I promise you, this is where I'm going to do a research paper, because, you know, all our muscles do weaken as we age, but you can work your skeletal muscles and keep them in pretty good shape. Maybe we can. Are you serious? That's crazy. There's no way that's 7 years old. He's got, 87. I was like, 7 years old. He's got to be on steroids. 87. Yeah. Yeah, I just saw a guy on Tinder the other day that was like 55, but absolutely ripped. Like we almost had an accident because we were driving back from Dothan. And Leslie, you know, my snorting partner was driving, and it's funny because we had her parents in the car too, which usually I don't like to ride with parents because I'm uncensored. But I let her parents come. Her parents were with us, and so I usually swipe when we're on the road because that's the most entertaining thing to do. It'd be like, oh, look at this idiot, you know. And then so I was like, whoa! I was like, look at this guy, he's 55, and I show it to her. And her mom is sitting right behind me, and it's just like the sweetest little. Her mom's sitting behind me, and she said something like, "Oh, is it another naked picture?" Because on Sunday, some dude somebody had sent me a picture that, and when I get a picture like that, I share with my friends, right? So I had sent it to my 3 tennis girlfriends, and her mother was with her, and she showed her the picture. Yeah. So I found that out on the ride back. I'm like, oh, you showed your mom? She was just like, oh, giggling in the back. I'm like, okay, no. And then she's like, no, Mom, this one's wearing shorts, kind of. But yes, you are right. You can work those muscles out, and I have seen it. Yes, there are people. Yeah, I mean, but so my idea for research is, can you work the iris out? Because it is a muscle. It's smooth muscle, though. It's not skeletal muscle. I don't know if you can't really strengthen smooth muscle. Well, there's a reason you can't strengthen smooth muscle, because skeletal muscle has the ability to hypertrophy. Smooth muscle doesn't. But maybe there's something we can do about it. I don't know. Anyways, I think it's pretty cool. So yes, your iris does weaken. You will begin to lose, not lose, but it'll be slower. Your response to light changes will be slower. So accommodating to darkness or accommodating to lots of light will be a little bit slower. Okay, and then your retina. Let's talk about the retina. It's just all jacked up. Your retina's messed up. It's gonna happen, right? All sorts of things, all sorts of things could happen, from macular, geez, why are you not? Oh, it's not on pencil mode anymore somehow. We almost made it, right? We almost made it with no hiccups. Apparently your ability to pinpoint your finger is gone too. All right, so retinal diseases, what are we talking about? Things like macular disease or degeneration. What's the macula? Somebody tell me. It's the center of the retina. Thank you. Macular degeneration. Detached retina. Remember how we talked about the vitreous body, how it holds the retina in place? Okay, so if everything that's being produced by your body is decreasing, that vitreous may also decrease, and that retina may detach more easily. What about glaucoma? Well, what is glaucoma? Glaucoma is a high, right, what? Right, high fluid pressure in the eye. So our eyes, that aqueous humor that's in that anterior chamber is constantly being recirculated, absorbed, and remade. If that angle has a blockage in it, You're going to be making aqueous humor, but no way to drain it. That's going to increase that intraocular pressure. And we don't want to do that, right? Because you have some very sensitive things going on here. Number one, you increase the pressure, that curvature of the cornea may change. Number two, you're increasing pressure in an enclosed space. You may end up cutting off some of that blood supply, which is really detrimental. So that's glaucoma. Cataracts, which is very common in the elderly. So as we grow older, not only does our lens lose its ability to accommodate, become less pliable, it may become cloudy or opaque. It may have some defects inside of it. So anything that's not clear happening in the lens is a cataract. Okay, it could be like a deposit. It could be an overall cloudiness. This next one here, it says about the age of 60, 25% of us will have a noticeable hearing loss. I promise you, those numbers are not right if we did the study today, right? Because how many of us spend at least half the day with our little earbuds in? Are they earbuds or earbuds? Every time I say it wrong for some reason. My daughter yells at me. What is it? AirPods, thank you. Because I've called them ear pods, I've called them earbuds, and she keeps telling me it's air something, so it's AirPods. Okay. We stick things in our ear and we turn up our music really, really loud. I know it because, well, first of all, I can hear y'all when I'm in traffic. And second of all, I know you hear me when I'm coming in every morning too, because that's how I like to hear it. And so, and that's why I'm so loud, because I can't hear. But yes, it does progressively go down. And I think it's a lot higher now because of all of this headphone usage that we've got. And not only that, but we talked about this. Did we talk about this in this class? How our bodies are changing from generation to generation? Yeah. So this newer generation that's coming up, and most of y'all are in this generation that have been doing this, you're losing the curvature in your neck. Yeah. Yes. And so now they're finding that kids, even at a younger age, are beginning to develop this loss of curvature in the neck to the point that we are actually changing the way human beings will be looking. Yes. So if your grandchildren are born like this, don't be surprised. Yes, it's your fault. Exactly. It's your fault. Yeah, I know. It's crazy. Look it up when you get a chance. But there's been several studies. Huh? Mm-hmm. It is freaky because we don't realize it. We don't realize it, but it's happening. Yeah, our bodies are changing because of it. I know, right? Okay. And then, of course, there's tinnitus. What is tinnitus? That is ringing in the ear, regardless of what caused it. Tinnitus becomes more common as we get older. Again, I'm really thinking this needs to be changed to like 40 at this point. Let's just put everything on 40. Why not? Do it. All right, so that concludes our special senses. Let's run through these PowerPoints to give you peace of mind, although honestly, I've cut them down so much, I'm just going to tell you to study everything anyways. And then we can do Kahoots. See what? This is recorded, so you don't have to take notes if you want to. What? Yeah, isn't that crazy? Every year? Do they get to go on disability if they don't pass the test? Oh. Oh, and they have to adhere to that. Yeah. Okay, when it comes to special senses, you got to make sure that you understand the names here. Olfaction is the sense of smell. Please understand the cells that are involved, your olfactory receptor cells, your supporting cells, and then your basal stem cells. And then when it comes to gustation, please understand gustation is taste. Know your 5 tastes: sweet, sour, bitter, umami. And then know your cells, your supporting cells, your receptor cells, your basal stem cells. Know that you have taste buds on the soft palate, the pharynx, and the epiglottis, along with some papillae. Which papillae are they? You should know the valate, the fungiform, and the folate. Okay? Vision. You should know the muscles, whether they're palpebral or extrinsic, what they do. The conjunctiva, where is it? What does it do? The lacrimal apparatus. The fibrous tunic is made up of the cornea and sclera, and the vascular tunic, the choroid, ciliary body, and iris. The right here, this entire slide is super important. You need to know what the iris does, but you need to understand that when the pupil is constricted, it is a circular muscle that is contracted, and that is done through parasympathetic. When the pupil is dilated for dim light, it's the radial muscle that's contracted, and that is the sympathetic system that does it. Please make sure you know what the blind spot is, what the macula is, and what the fovea is. Your photoreceptors, rods and cones, you should know what they are for and what their photopigment is. Okay? Vision, understand an emmetropic eye, astigmatism, a myopic eye, and a hyperopic eye. Understand binocular vision, and this part right here. Hearing and equilibrium, you should know the different parts of the external ear, middle ear, and internal ear, and what each one does, their jobs. Ceruminous glands are going to secrete earwax, right there. External auditory canal is going to transmit sound, auricle captures sound, and the tympanic membrane vibrates with that sound. And then your middle ear, you should know the importance of the auditory tube or the eustachian tube. And then you got your three bones. Inner ear, know it all. You already know it all. Don't worry about that. Okay. And then for aging, actually, I do want you to know presbyopia and cataracts, glaucoma, and tinnitus. Okay. Let's go to the senses. I'm very quickly becoming one of those weird, like off-ball old professors. Go ahead and do autonomic. So this should be like a review. You guys should already be. There's two more. Are you serious? There's two more. It's not spring break anymore, y'all. Wake up. Not spring break anymore. It's over. And senses. Mm-hmm. All right, for the autonomic system. Yes, there's 3 PowerPoints. Autonomic, you should know what a preganglionic, what a postganglionic is. This is essential. You guys already know that. Rest and digest, fight or flight. You don't have to worry about that. You should know a preganglionic motor neuron and a postganglionic motor neuron. When we're talking about the divisions, you know that sympathetic is thoracolumbar, short preganglionic, long postganglionic. The parasympathetic is craniosacral, long preganglionic, short postganglionic. The trunks, you should know the sympathetic has either sympathetic trunk ganglia or prevertebral ganglia. The parasympathetic is all what? Terminal ganglia. Terminal ganglia. That's me writing terminal. Okay. Please make sure you know all 4 options of how a preganglionic axon of the sympathetic division will synapse or can synapse with a postganglionic neuron. Am I going too fast? Sean, am I going too fast? Okay, because you're actually writing. Okay. Dude, it's like everything. You might be better off just writing the slides that I skip. Okay. For the autonomic plexuses, celiac is major. You should know what that innervates, and then the rest you can make up just by knowing the arteries. Superior mesenteric was the small intestine. inferior mesenteric, large intestine, renal, kidney, cardiac, heart, pulmonary, lung, hypogastric, pelvis, organs in the pelvis. Okay, and then the neurotransmitters and the receptors. Please know what cholinergic neurons secrete and what their receptors are, and then know what adrenergic neurons release. Know which one is which. So all of this here you should know, cholinergic and adrenergic neurons. When it comes to the receptors, be able to recognize that cholinergic neurons are nicotinic or muscarinic. When it comes to your adrenergic receptors, this is your bonus for the test. Okay? So for your bonus, you should know that alpha-1 adrenergic receptors are found in smooth muscle fibers in blood vessels. This is what you can write, Sean. Write this down, man. It's your bonus. Alpha-1 smooth muscle fibers in blood vessels. I'm going to slow down because I know some of you are writing this until you hear it. Okay. Alpha-2. Alpha-2. Cells of the pancreatic islets. Why? Because that's important. Insulin secretion is important. Okay. Beta-1. Oops. Sorry, guys. Beta-1, cardiac muscle. And juxtaglomeralar, but if you just remember cardiac muscle, it'll have both of them. Okay. Beta-2, smooth muscle of the airways. Smooth muscle of the airways and blood vessels of the heart. And again, when it has two things that are important, as long as you can recognize one of them, you'll be able to match it. And then beta-3 is your brown adipose tissue. Keeps us warm at night. Beta-2, smooth muscle of the airways and blood vessels to the heart. Do what? Okay, so everybody got the bonus part down? All right. Okay, when it comes to this chart, the sympathetic and parasympathetic, if you want to skip these 8 pages and just the effects and all that, sorry, this chart you can read through. We already did the PowerPoints to it, but then when it comes to the effects, you can skip these 8 slides if you want to just do it from the one sheet that I put on there, and I'll show you where that sheet is, okay? I'll show you at the end. I put it right under the PowerPoint. There's a one sheet of chart of all the effects that I put on there. Okay, as far as integration and control, you already know what a reflex arc is. I hope you should know what autonomic tone is and understand what the hypothalamus, or that the hypothalamus plays a major role in integration with the autonomic nervous system. Okay, let me show you the chart. It should be right after that PowerPoint. You should come up on it. Ooh, hold on now. Am I on the wrong one? It'd be so funny if you guys were on class I didn't post it for. Ah, right there. So if you wanted to skip those 8 slides about the effects, you could simply just go to this chart right here and look at that. Okay? Tried to make it easy for you guys. Don't say I don't give you no study guides. There you go. You got a study guide. All right. What did we just do? Autonomic? We didn't do senses yet, right? Okay. We did senses? No. Okay, so senses. Yes, obviously know the definitions here, sensation and perception. And then know your general senses and your special senses. Be able to categorize them, whether somatic or visceral, so you should know all that. And then you want to know, well, you do want to know this at free nerve endings. These two slides here, just the top line, this part, and that, well, you know. Okay, and then I'm going to go right to the chart because that's where it's at. Exteroceptors and interoceptors you should know, and proprioceptors. Be able to classify them as that. Know what an exteroceptor is, what an interoceptor is, what a proprioceptor is. And these charts will help you do that if you want. Right here, exteroceptors, interoceptors, proprioceptors. And then this also, mechanoreceptors detect mechanical stimuli, thermoreceptors, thermal temperature changes, nociceptors, pain, stimuli, photoreceptors, light, chemoreceptors, and osmoreceptors both detect chemical stimuli. So know these receptors and the stimulus that they detect. Okay, and I already told you, you should know your somatic sensations, your tactile sensations. These here. Know fast pain, slow pain, the difference between the two, and when you see the two, whether it is somatic or visceral. Proprioceptors, you know what they do and know what they are, the muscle spindles, the tendon organs, and the joint kinesthetic receptors. Okay, and this is just a review of all of that. Pathways, understand a first, second, and third order neuron. Where do they carry things from where to where? I would do it from this page right here. First, second, and third order neurons. And that is it. Any questions?